This video is going to take you through the first bit of Chapter 9, which is the Muscle Physiology chapter. This chapter can be very difficult for students to understand because there are a lot of things moving in a bunch of different directions, so it can be very helpful to watch some videos about this. I'll link to some more than likely in either Schoology or YouTube for you guys to watch, but I recommend that you watch a few videos to help you understand what's going on through here. First off, there's a slew of terms for you guys to define. They're pretty self-explanatory, and some of it we've already talked about before. So voluntary and involuntary, we hit back in tissues. Muscle fibers are defined in that first sentence. They're just the same thing as cells. After that, you have your three different types of muscle tissue, compare and contrast. We really did this back in tissues as well. What I'm wanting you guys to focus on here is your highlights for your types of tissues that we talked about back then. So you should know that this is skeletal muscle because the fibers do not branch, they are striated, they are multinucleate, and so those are your characteristics I want you to focus on for skeletal muscle. You should recognize this one as cardiac muscle because it's striated just like skeletal, but some of the stripes are extra dark. Those are intercalated discs, and only cardiac muscle has that. Also, the cells branch off and they go in multiple directions. The nuclei usually appear to be smushed to the very outside of the cell, and usually each cell just has one nucleus to them. Smooth muscle can look a lot like some of our connective tissues, especially dense regular connective tissue, but you need to remember all of this is cellular. There's no extracellular matrix here. The cells are fat in the middle and then taper off towards either side. They are not striated. There is just one nucleus per cell. What are your four special characteristics for muscle? So every edition of your book, they either add a characteristic or they take one down. So my notes might not necessarily correspond with the number that you're supposed to have. Sometimes your book likes to combine some of these, but you do need to know that skeletal muscle is responsive. If you stimulate it, it will do something as a direct response of that stimulus. Skeletal muscle is contractile. It is the only tissue in your body that can forcibly contract. So when it does that, the fiber is generally speaking going to get shorter and fatter and then move something as a result of that contraction. Muscle is extensible and it's elastic. That means you can stretch it, and after you stretch it, it will come back to its original size. Functions for muscle. It kind of depends on which kind of muscle you're talking about, because really all of them, they're movement. Sometimes we move you, sometimes we move stuff through you, but we're going to be moving stuff around. As we do that movement, there are some other things that are important that happen. We talked about muscle helping you to maintain the alignment of your joints. Well, that also means it's going to help you maintain your posture, because posture is about maintaining the joint alignment in your vertebral column. And so maintaining posture, that's part of this. Again, stabilizing joints, that's all the joints that aren't your vertebral joints. They can also help you to generate heat. Hopefully you guys have noticed when you've been working out, you get hotter. Explain the nerve and blood supply to each muscle. So each skeletal muscle has a nerve that runs to it so that you can tell it when it's time to contract and when it's not. There's at least one artery that goes to it. If it's a really large muscle, it might have more. And then there's multiple veins that drain the blood from each muscle. Next up, you have describe the connective tissue sheaths. This is an old picture from an older version of the textbook. So this is a muscle cell. It has a sheath around it, and then multiple muscle cells get bundled together to make a structure called a fascicle. There's a connective tissue sheath around a fascicle. Then a bunch of fascicles get bundled together to make the whole muscle. The whole muscle is surrounded by a connective tissue sheath called the epimysium. So I don't remember if this is pork tenderloin or something going on with a cow, but if you notice, this is a whole muscle that came out of that animal, and you can see the connective tissue on the outside of it, so the epimysium is still present on this tissue. I will also mention plenty of people would prefer you say epimysium. It just kind of depends on who you're working with, but I'm going to say epimysium because I'm a Texas girl, so there we go. Paramecium is how you separate a fascicle. So this is a fascicle. It has paramecium around it. Here's a different fascicle. It has paramecium around it. Then each cell has a sheath around it, and not only does it have a plasma membrane, which isn't called that anymore, it's going to be called a sarcolemma, but outside of the sarcolemma we have endomysium that goes around that. So there's endomysium around that cell, there's endomysium around that cell, there's endomysium around that cell. All of those connective tissue sheaths are going to merge together at the end of the muscle to make the tendon that helps attach that muscle to bone. Origin and insertion. Each muscle has a couple places where it's going to attach, if it's attached to the limbs anyway. Some of your muscles do attach to skin and they work differently, but for the most part, We have an origin, which is where the muscle attaches to the bone that it's not going to move. And then we have the insertion, where it attaches to the bone or bones that it is going to move. So this is the biceps brachii. The origin is up on the scapula. The insertion is down on the radius and the ulna down there. Tendon is dense regular connective tissue that attaches a muscle to bone. Aponeurosis is a sheet of connective tissue that attaches a muscle to another muscle. This is the occipitalis, this is the frontalis. The Galea aponeurotica, it covers the surface of your head underneath the skin. It's the aponeurosis that connects those two little patches of skeletal muscle to each other. Next up, sarcolemma. It's the plasma membrane of a muscle cell. One of the main beefs that student has with this chapter is that everything has to change names because muscle behaves differently from some of the other cells that you have in your body. So instead of cytoplasm, we have sarcoplasm. Instead of the plasma membrane, we have the sarcolemma. There's just a lot of term changes, and so make some flashcards and that can help you out with those, because if you don't remember what these are, you're never going to be able to answer the questions that relate back to this. This is a chunk, one piece of one muscle cell. So this is the sarcolemma that's been peeled back in this spot. Because this is a skeletal muscle cell, it is multinucleate. So there's a nucleus, there's a nucleus, there's a nucleus. There's mitochondria present inside of these cells. And then the rest of the cytoplasm, which is now called sarcoplasm, is packed with these structures that are called myofibrils. These two things are going to help muscles store chemicals that are necessary for the muscle to be able to do their job. So glycosomes, if you break down the glyco part, it means sugar. Well, glycogen is how we store sugars inside of our cells. We keep some of that sugar stored inside of our skeletal muscles so that skeletal muscle will have a fuel source that is readily available to it. Myoglobin, this means muscle. This globin is kind of like hemoglobin. So this is kind of like hemoglobin, but in your muscles. So where hemoglobin transports oxygen around your body, this stores oxygen in a muscle cell. So I know that at some point in your life you've heard the term dark meat and white meat, or light meat, whichever one you called that. The difference in these two things has to do with how much myoglobin is present. Myoglobin is a dark pigment, kind of like hemoglobin is, and so the more myoglobin present in a muscle cell, the darker that cell is going to appear, which means the darker the tissue as a whole is going to appear. So this, I'm going to assume that this is turkey. It looks like turkey on the other side. And so this has more myoglobin in it. This has less myoglobin in it. That means this muscle will be able to contract for a longer period of time without tiring because it has oxygen stored. This is going to get tired a lot faster and run out of energy because it didn't have any oxygen stored. Myofibril. So I pointed those out a slide a couple of pictures ago. This is one chunk of one myofibril. So just to come back to that slide, this was the cell, aka a fiber. It's packed with all of these things called myofibrils. Coming back to this is just part of that myofibril. So this is mostly what's inside of a muscle fiber. So notice where it says it accounts for 80% of the cell volume. This is the part that does the work. So most of the cell is made out of this. So there's this zigzaggy line right here. It's called a Z-disc. There's another zigzaggy line right here. That's another Z-disc. This, from one Z-disc to another Z-disc, is called a sarcomere. So this is a sarcomere. This is the next sarcomere. So I tend to like to consider myofibrils as chains of sarcomeres. So it's just sarcomere, sarcomere, sarcomere. Each one of these is the contractile unit, and so each one of these can forcibly shorten to move something. Striations. Now, because of the very regimented alignment of the filaments, either the thick filament or the thin filament within a myofibril, you end up being able to see stripes on the tissue as light stripe, dark stripe, light stripe, dark stripe. So the striations that you saw on skeletal muscle and cardiac muscle, they occur because of this structure that's going on through here, and you end up with light stripe, dark stripe. I am not sure how well I'm going to be able to doodle this on this screen, but I'm going to do the very best that I can. Here we go. So, underneath all of these little terms that we have, I gave you some room at the bottom of the page to doodle some stuff. We're going to doodle a sarcomere right here, and we're going to label one a little bit later on. But a sarcomere goes from one zigzaggy line called a Z-disc to the next zigzaggy line called a Z-disc. So we'll label that Z-disc. And then this is the next Z-disc. The whole thing is called a sarcomere. Okay. There are a couple of different proteins that make this up that are going to matter for us. Hold on, change colors. I gotta back out. Discard. Oh man, and now it just deleted everything. Okay, give me a second to get back in to where I was and redoodle some things. I hit escape too many times. Okay. You guys already have your labels, so keep them. Z-disc, Z-disc, whole thing as a sarcomere. Okay. This time I won't hit it too many times. Next. Coming off of that Z-disc, you have some, well, we're gonna ignore the elastic filaments and the titin. So we're just gonna draw some filaments that are gonna come in towards the middle of the cell, but not go all the way to the middle. And then there's going to be some fibers that run towards what we just doodled from the other side. These things that we are doodling on here right now are called thin filaments. They are primarily going to be made out of the protein actin. But there's going to be some other regulatory proteins that are going to be found on there. Okay, I'm going to switch colors again. Next, we're going to get some more proteins that go like this through a sarcomere. These are called thick filaments. What's special about them is they get these little heads on them. So that little thing that I just drew is one head. What the heads can do is they can grab onto the thin filament and then pull the thin filament closer to the midline of this sarcomere. So the thick filaments have heads, the thin filaments have places where those heads can grab. Next, we have a bunch of different components to this that we are going to label, and they're the next terms that come in your notes. So, first off, you have an A band. The A band goes from one end of the thick filament all the way to the other end. So this is the A band. The I band is the place where there is only thin filament. Z-disc is going to be right in the middle of the I band. Now, I only drew one sarcomere, but there would have been another sarcomere over here. There would have been another sarcomere over here. So there would have been a little bit more to this I band on the other side, which is why I extended that bracket a little bit farther. Next up, we have the H zone. That is the place in the middle where there is no thin filament. Oh, it deleted my stuff. So right there, H zone. Right in the middle of the H zone, there's a little, your book depicts them as 3 lines that run down the middle like so. That's the M line that runs straight down the middle. Now we already did the Z discs. The red lines were the Z discs. And then this whole entire thing is a sarcomere. So this is the part of a muscle cell that actually carries out the work of contraction. The way that it's going to do that is the head of a thick filament is going to grab on to the thin filament and then pull it closer to that M line that's in the middle of that sarcomere. Okay. Making sure we got all that in the notes. I'm going to go really fast because I said all of this. Remember, you can pause the video to get stuff. So there's your definition for A band and I band, definition for H zone, definition for M line and Z discs, and then your definition for a sarcomere. So I said all of that earlier, we're good. So here's the picture from your book that shows you what we just talked about. So they've even labeled the I bands, the A bands, they've labeled the H zone, and then the M line is that set of 3 lines that go straight down the middle of the H zone. Next, myofilaments are the thick and the thin filaments that make up the anatomy of that sarcomere from just a second ago. The thin filament, well, it does have some other stuff. It's going to mostly be made out of the protein actin. And the thick filaments are made out of the protein myosin, and that's where it has the heads that can grab, which are sort of these little extensions that are coming off right here. After that, I told you to be able to recognize various parts of a muscle from macro to microscopic. So this picture is to show you those connective tissue sheaths again, where we have the epimysium, the paramecium, the endomysium going around one muscle cell. You can see the muscle cell has a bunch of myofibrils. They didn't label that, but if you wanted to add it, that is one myofibril, which is where the sarcomeres are going to be found. Next, I gave you this picture to label. They've already got the filaments, so the thin and the thick filaments are already labeled for you. They did go ahead and show you the elastic filaments that attach the thick filament to the Z-disc. I left those out of my picture, but they do exist. The Z-disc to Z-disc thing is a sarcomere. What's missing from this picture are all those bands and zones that we had labeled earlier. So just to make sure you have those again, here to here is the I band. Here all the way across to here is the A band. Here to here is the H zone. And they already have the M line labeled for you guys. Okay. I'm going to post a quiz over kind of what we've just talked about to make sure you understand everything that we've gone over up to this point. So make sure you look for Schoology so that you can take that quiz. Okay. Here's a video to show you how everything is going to interact.
We move our bodies by shortening or contracting our muscles. Let's take a closer look. A muscle consists of parallel muscle fibers. Each fiber is a single cell in close contact with a motor neuron. Each muscle cell contains bundles of parallel myofibrils, shown in red, surrounded by endoplasmic reticulum, or ER. An action potential traveling down a motor neuron initiates an action potential in the muscle cell. The action potential spreads along the membrane and down tubules that extend into the cytoplasm, causing the myofibrils to contract. Each myofibril consists of a series of sarcomeres arranged end to end. The sarcomere is the contractile unit of muscle. Each sarcomere consists of thick filaments of myosin, shown in purple, and thin filaments of actin, shown in orange. A sarcomere contracts when its actin filaments slide past its myosin filaments. Contraction shortens the sarcomere, but does not change the length of the actin or myosin filaments. The myosin filaments have heads that bind and pull actin repeatedly, shortening the sarcomere. How do the myosin heads pull actin? In a relaxed muscle, the myosin binding sites on actin are blocked by a protein complex, and ADP and phosphate are bound to each myosin head. An action potential causes the ER to release calcium ions, which bind to the protein complex, causing it to shift and expose the myosin binding sites. The myosin head binds to actin. When ADP and phosphate are released, the myosin head bends, pulling the actin. This is called the power stroke. Next, ATP binds to the myosin head, causing it to detach from actin. When the ATP is broken down to ADP and phosphate, the myosin head extends. The sequence repeats as long as calcium ions are present. Bind, power stroke, detach, extend. The combined work of many myosin heads causes the actin filaments to slide past the myosin filaments. When the action potentials stop, calcium ions are pumped back into the ER. The myosin binding sites on actin are again blocked, and the muscle relaxes. During muscle contraction, actin filaments are pulled toward the center of the sarcomere. The simultaneous contraction of the sarcomeres shortens the myofibrils, and the entire muscle cell contracts. This is how a muscle cell responds to action potentials in the motor neuron. When many muscle cells contract together, the result is movement.
All right, hopefully that gives you an idea of at least the parts that are going to be in play. We'll talk about this more in the next video too.